Hi, my name is Dr. Mhairi and I am a Senior Lecturer in Biomedical Science here at Montfort University. And I'm passionate about cancer research. I'm particularly interested in the link between certain viral infections and how they can cause cancer. In 2012, there were over 160,000 deaths caused by cancer in the UK alone. This equates to about 29% of all deaths recorded in the UK at that time. In a recent article in the British Medical Journal, former editor Dr. Richard Smith glibly remarked, "Cancer is the best way to die, and that we should stop wasting billions trying to cure cancer," sparking outrage amongst cancer survivors. Contrary to Richard's idea that cancer is only unpleasant in the last few weeks, Larry, a 59-year-old advanced pancreatic cancer patient, spells out the many and varied ways that cancer is extremely unpleasant throughout, psychologically, physically and emotionally, and re-emphasises the importance of funding cancer research, which has dramatically increased the survival rates for many types of cancer. So what do we do here at DMU, and why is it important? Well, first, a little background on what different cancers have in common. We call these the hallmarks of cancer. Broadly speaking, cancer is a genetic disease caused by one or usually more mutations in the DNA. These mutations can cause a cell to acquire a number of properties that give it a survival of the fittest style advantage. Most commonly, these mutations lead to an increased rate of growth in cancer cells or the ability to ignore the normal signals that tell them to stop growing. Often, cancer cells will acquire numerous mutations that enable them to take advantage of many of these common properties, the most deadly of these being invasion and metastasis. That is, the ability to pack up its tent, move, and then set up camp elsewhere. Around 90% of cancer-related deaths result from the secondary metastasis and not the primary tumour. Think about that for just a minute. 90%. That means that of the 160,000 or so deaths in the UK in 2012, statistically speaking, 144,000 of those would have been a result of a secondary tumour. So if we can halt this process, think what this would do for survival rates. My own research interests lie in the role that a certain virus-encoded cancer-causing protein plays in making the cells it inhabits more mobile and invasive. It is estimated that around 15 to 20% of cancers are linked to a viral or bacterial infection of some description. Amongst these is Epstein-Barr virus, or EBV, which is more infamous as the cause of glandular fever. Now, whilst the majority of us carry EBV as a dormant virus in our tonsils or lymph nodes, only a very small proportion of us will go on to get cancer, and even then it is more likely as a result of other environmental factors as well. However, important lessons can be learned from this experimental system, lessons that can be applied to most cancers, in fact. So how does this seemingly harmless little virus wreak so much havoc? Well, it's all down to a protein called latent membrane protein 1, or LMP1 for short. LMP1 is what we call a transmembrane protein. That is, it sits within the plasma membrane that surrounds our cells. Inside the cell, LMP1 has a long tail which has two signalling domains. These are called CTAR1 and CTAR2, which simply stands for C-terminal activatory region. In other words, it activates cell signalling. It is well known that these signalling regions activate numerous pathways that are often deregulated in cancer, such as those involved in promoting survival, enhancing cell motility, and matrix deposition. But something I've been looking at recently is the ability of this protein to activate a new signalling pathway in order to make the cells it inhabits more mobile and invasive, and this suggests it might be important for causing a primary tumour to metastasize to a secondary site. By activating signalling from a family of pro-inflammatory proteins called cytokines, LMP1 is able to make epithelial cells throw down a matrix on which to travel across, much like this scene from Wallace and Gromit, The Wrong Trousers, where Gromit runs out of train track and picks up some spare track, throwing it down in front of him so he can continue to travel on his path. This in turn makes the cells more motile because they have a track on which to travel. They will move more quickly as a result. LMP1 also causes epithelial cells to spew out a family of proteins called matrix metalloproteinases that chew up the proteins in the environment underneath them. In the same way a machete allows a person to hack his way through the thick undergrowth in the jungle, These metalloproteinases allow the cancer cells to cut their way through the basement membrane and gain access to the blood vessels, along which they will then travel to their new destination. Now this is all well and good, but unless the cancer cells can survive long enough to reach their secondary site, they won't be able to establish a secondary tumour. In order to do this, they provide themselves with a potent pro-survival signal, much like an elixir of life. This buys them enough time to travel to their new site, be that the lungs, the liver or the brain, and to set up a new camp where they will grow and divide and create a new settlement. By growing epithelial cells that express LMP1 in the lab, we can play around with different treatments, different drugs, different environmental conditions, and then see what happens to the cells. And this helps us to understand how cancer grows, how it evolves, and how it moves around, giving us clues as to what we can do to prevent it from spreading further.